My name is Michael Hunt, and I will be presenting on bioprinting and bioprinting technologies. This presentation is for Concepts of Biology through the University of Maryland Global Campus. Let's get started. Over the course of this presentation, we will define bioprinting, the applications of bioprinting, the limitations of bioprinting, the types of bioprinting, what stem cells are and their role in bioprinting, what bioink is, and what the future holds for bioprinting. Let's start off with what is bioprinting. Bioprinting is a technique of creating bones, organs, tissues, and cartilage, really anything that the human body contains, out of organic material via 3D printer. Just like in 3D printing, a material is fed through a nozzle to produce a structure, starting layer by layer until the structure is complete. This is done by creating a digital 3D image and uploading it to the printing machine. There are four main types of bioprinting methods: inkjet, extrusion, laser-assisted, and stereolithography. We will later go over what each of these methods are and what their advantages and disadvantages are. What are the possibilities with bioprinting? Over the years, bioprinting has been attempting to create new body parts for those in need. Bioprinting hopes to create things like skin for skin grafts and new heart valves, and replacing synthetics. The main advantage to bioprinting comes clear when talking about organ donations. Making an organ out of someone's own cells greatly reduces the odds of rejection of new cells that would be in the body versus a donor's organ. Next is the limitations of bioprinting. Although bioprinting has been around for nearly 20 years, not everything has been ironed out. When it comes to creating complex structures like brain structures, the neurons in these structures are incredibly complex and challenging for the current technology that we have. In cases where cells are not native, the odds of a transplant being rejected go up tremendously. This is the case sometimes for bioprinting. And also, often the structures developed by bioprinting degrade and break down rapidly. Now we're going to go over the different types of bioprinting. First is inkjet printing. Inkjet is the first type of bioprinting created and is most similar to PLA 3D printing. It uses a container of bioink, which is pushed through a nozzle, where it creates a layer by layer formation to create the structure. The main advantage of using the inkjet method is that it is one of the cheapest methods and has a high cell viability. A big disadvantage is prolonged usage can create damage within the parts. Next is extrusion printing. Very similar to inkjet printing, it is similar in the way of how it creates layer by layer via nozzle, but is different in that it uses a continuous cylindrical stream onto a printing surface. This gives it a higher control over creation of the structure. A downside to this is it has lower self-viability than the inkjet method. Next is laser-assisted printing. Laser-assisted printing is a method that utilizes a laser to direct where the cells go in the bioink. There are two main advantages to using laser-assisted printing over the other methods. It has a higher self-viability at around 95% and a higher resolution on the detail of the structures. The big downside to using laser-assisted printing is the high cost, and there's little data to show for what the longevity of created structures will be. The last method that we will be going over is stereolithography printing. This is very similar to 3D resin printing, and it utilizes UV light to solidify the structure that is being created. Just like other forms of bioprinting, it uses bioprint as the medium in which it creates the structures. The advantage of stereolithography are fast printing speeds, high cell adhesion, and extremely high cell resolution. The downside to using this method is the high cost, and the cell viability can lower due to UV exposure. Now let's talk about stem cells and their role in bioprinting. Stem cells are the main type of cell used in bioprinting, and it is for a good reason. Stem cells can be used to replicate numerous types of cells, which make it the workhorse of bioprinting. When stem cells are created from somatic cells of the recipient of the new structure, the odds of the structure being rejected go down drastically. Let's talk about what we've been talking about this whole time: bioink, the thing that actually makes the structures. Bioink is the base of all the structures made from bioprinting. It is made from a polymer and a cell. These specific components of bioink vary depending on the need of the project. Fundamental structure stays the same over what the object actually is. There are many advancements coming out in bioprinting technologies. One interesting advancement is in pseudo bioprinting, or essentially printing inside the patient's body. This is less invasive than full-fledged transplant surgery, and this method uses the body's recipient as the bioreactor of the new cells, increasing the chance of a successful procedure. Nanotechnology is another extremely advanced way of bioprinting. This technology is using nanotechnology that has implications of creating new structure with high cell viability. These new technologies can create highly complex structures, which can fix some of the problems mentioned at the beginning of the presentation about complex structures. What does the future hold for bioprinting? The advancement of bioprinting is expanding further and further. It will one day be commonplace in hospitals around the world. Experts theorize that over the next 2 decades, bioprinting will be fully capable of amazing things. Thank you.